section, I'll be talking about electron transport chain and oxidative phosphorylation. But first, let me tell you, what does oxidative phosphorylation mean? It is a metabolic pathway in which cells use enzyme to oxidize nutrient, thereby releasing the chemical energy stored within in order to produce adenosine triphosphate, also known as ATP, a molecule that carries energy within the cell. This takes place in the inside of the mitochondria membrane. On the other hand, the electron transport chain is a series of electron transporter embedded in the inner mitochondria membrane that transfers electron from NADH and FADH2 to molecular oxygen. But what are the process of electron transport chain? Let's find out. All eukaryotic cells, from yeast to those that make up the human body, contains membrane-bound organelles with specialized function. Mitochondria are doubled membrane organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from the reaction that takes place at the inner mitochondria membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing the concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called F1F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. This ATP molecule ultimately provides the energy for most of the cell's reaction. The synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no protein gradient, synthase subunits stop rotating and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are four protein complexes, numbered 1, 2, 3, and 4. Complexes 1, 3, and 4 directly pumps protons from the matrix into the intermembrane space. Complex 2 does not directly pump proton, but it does promote proton pumping in complex 3 and 4. Proton pumping requires energy, and the four protein complexes get this energy by transferring electrons through a series of coupled reactions. This link process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they pass along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electron based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron to jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to coenzyme Q molecule. Complex 2 is similar to complex 1 in two important ways. First, high-energy electron also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfer electron between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donates their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through two redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving two electrons convert a molecules of oxygen to two molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. Now that I've discussed all the process of electron transport chain, let's compute what is the total number of ATP generation per glucose molecule after its complete oxidation. 38 ATP molecule can be made per oxidized glucose molecule during cellular respiration, 2 from glycolysis, 2 from the citric acid cycle, and about 34 from electron transport chain. Thank you for watching. I hope you learned something from this video. This is Eliza RBD Operanya signing off.